Well, exactly. We are the team of the University of Calgary. May I introduce to you, this is Vlad and Patrick. These two are bioinformatics students of the Bachelor of Health Sciences program at our university. I'm Sebastian, I'm a PhD student of computer science. And in order to introduce you to our motivation of our iGEM project, I want to take you on a tiny detour to the realm of swarms. Swarms, as all of you are aware of, are usually seen in nature as schools of fish, flocks of birds, swarms of bees or ants, as seen in these two pictures. And by their great accomplishments in nature, they have motivated computer scientists to put through this paradigm of swarm systems in various disciplines of our research. I'll give you some examples of their great potential that we are using in computer science. One of them is depicted in these two images just by being aware of the local neighborhood. These tiny swarm dudes here, represented as red spheres, can find the peaks of mathematical functions, thereby optimizing, solving optimization problems. With a more complex set of interaction rules, they can the phenomena that can occur can be, well, more complex by themselves, and prominent examples of these emergent phenomena that we call them are the artificial immune system, or, well, in the real world, the immune system of the immune response of the human body, or gene expression as seen in the right picture here. Emergence, I have to mention that. We call usually those phenomena emergent phenomena where the systems that these swarm dudes form and build up show attributes that none of these swarm individuals by themselves possess. Another great approach is using the great creative capabilities of our swarm individuals by allowing them to build up three-dimensional structures in space, as seen here by these two pictures, or by this picture composition and this one image there. But if we reduce it to only 2 dimensions and take the liveliness of these swarm agents and add some color, we already get great capabilities as well. They are very entertaining and allow for lots of interaction. And that led us basically to what we wanted to do for our iGEM project. So Sebastian, what exactly does it take to put together a swarm? Well, for a swarm, obviously, as you have seen in all these simulations, we need high numbers of swarm individuals.
Well, that's easy to do with bacteria. We've got millions of bacteria on any given plate.
Yeah, but we also need interaction, as you have seen.
What do you mean exactly by interaction?
Well, all these swarm individuals have to be driven by certain urges.
Such as?
Well, in all of our simulations, these three-dimensional agents or swarm individuals had to have some movement of some kind.
Yeah, we can exert a certain control over bacterial movement. What else?
Well, any other kind of actuator would be welcome to make them more interactive.
Things like changing color or emitting a signal?
Oh, that would be great, just as we have seen in our last slide of these arts examples. Oh, sorry. And the final thing to realize these urges and interactions would be a set of sensors. Do we have that?
Yeah, we have a set of membrane sensors we can use with bacteria as well.
All right, then I, as computer scientist, I'm totally astonished, and I say, now the circle is complete and we can come back from programming back to life.
All right, so my name is Patrick King. I'm going to take you on a quick tour of our project. So the idea was based around engineering 2 strains of bacterial cells, both E. coli, a sender strain and a mobile receiver strain. At the simplest, highest level, the sender strains are activated by light using the light sensing system developed last year. And as a result of the light input, they emit AHL and aspartate as two signaling molecules. And our receiver cells will react to each of these in a different way. Getting into the sender cells a bit more, they are non-motile, so after putting them on the plate in a lawn, they will remain exactly where we put them. They are light activated, and as we said, they emit both signals, aspartate and AHL, to the receivers. The receivers respond to each of these chemical signals in different ways. They respond to aspartate by chemotaxis, as aspartate is a metabolite. This is a built-in feature in E. coli. And by AHL, we've introduced a biobrick system that causes them to glow, produce GFB. We also had to do some thinking about engineering the actual media mixture on our petri plates. In order to have the receiver cells swimming around, we needed to incorporate swim media into the plates, which is a low density media that allows the cells to move. And to keep the sender cells fixed in place, we also wanted to have a higher density media that would hold them in place. So we built them one layer on top of the other, the higher density regular media on top and the swim media and below. And this would also provide minimal obstruction to the light, which would be shown on the plate. And the sender cells will, of course, produce their two signals in theory, and the receiver cells will react to them. We constructed computer simulations of how this model should work. And this gets into also one of the goals of this project was to be able to design patterns of light, not just irradiate the entire plate, but to design patterns of light and draw out light shapes onto the petri plate. And hopefully we would see the bacteria assume those shapes once the signaling mechanism gets into play. The simulation was grid-based. Each grid contains concentrations of AHL and aspartate. And the interactions in the simulation were between the agents, the small green triangles you can almost see, and the red triangles are the senders. And it's also highly adjustable. As you can see on the left, we have a great deal of parameters. And the idea was that these parameters would be tweaked and adjusted as the simulation or as the experiments progressed. There are two visualization modes in this simulation, one for AHL and one for aspartate. They both diffuse throughout the petri plate fairly quickly. The aspartate not so quickly because the cells, as they are attracted to it, will also be consuming it. This is a quick video of one of the simulations. As you can see, the cells gather quickly close to the center where the senders are and turn green when they get there. And afterwards, they follow a slight, after consuming all the aspartate nearby, they follow a slight gradient away from the center of the plate. This is the same video again, but with the AHL in the visualization instead, which of course is not consumed, and so just diffuses everywhere very gradually. And this is the same visualization, or the same simulation again with the aspartate visualized again. So now we're going to get more in depth into the actual parts we built into our two cells. Each one would have two different biobrick parts that we assembled. On the sender, we would have a constitutive device for producing the light sensing system, which would be always on. We represent the light sensing system with the blue shape there. And as a result of the light input, this would activate the second device in the sender cells, producing both AHL and aspartate. Both these signals arrive at the receiver. AHL binds with LuxR, produced by a constitutive LuxR device. This is required for the signal to be received, of course. LuxR, another second LuxR device produces our GFP, and the chemotaxis system is built into the E. coli already. We designed parts for all of these and succeeded in building one or two of them, as you'll see later on. And we did an awful lot of planning and lab work, as you can see, or lab planning at any rate. We consulted with a great deal of people and received some really good help at the university. Right. So there you go. I'll let Vlad take over now.
All right, so I'm going to describe sort of the laboratory aspect and the work we did in the labs. Here's just some scanned images of our lab notes. We were trying to be thorough and document everything. So let's just get started here. Yeah, all right. So what we started out with is an assembly plan. We wanted to get an idea of where we needed to get. So we knew what we wanted for our final product. On the left there are the sender cells, which are the more complex construct. And on the right are the receiver cells, where you see the LuxR gene, as well as the GFP, which is induced by the presence of AHL. So at the top there you can see with the orange dots, those are the original parts from the registry that we'd be working with. And then here we have the intermediate components. Now yellow ones are just intermediates, but the ones with the blue dots are ones that we thought would be useful to submit to the registry at some point. And we have several levels of it. We did try to implement rolling construction because it's just the fastest way of doing it. And then another cycle, and finally we get our complete constructs. So on the topic of part assembly, of course it's extremely important to any project at iGEM. Now we started off doing standard assembly, where you have to isolate plasmids, digest them. What we found is that the yields of the plasmids were quite low, and using standard methods like mini and MIDI preps, we really were getting poor progress. It took us easily 3 weeks to get parts together. So we thought a better way to do it is with PCR. Now the advantage there is that we pretty much didn't have to do any plasmid isolation. We could use the plasmid sent to us from MIT directly as template for the PCR. We can generate as many parts as we want in parallel, so if you want 50 parts, you can do them all in the same reaction, well, in separate reactions, but at the same time in a thermal cycler. Like I said, it's extremely fast, because you can get all these parts within a few hours without waiting to grow up, you know, these 100 mil cultures without isolating plasmid, etc. And we had the idea of staying in DNA for as long as possible, because after you ligate some parts together, they still retain the primer sites, and so you can PCR amplify that again, and then do another digest, another ligation, and then amplify that again. So you don't have to worry about sort of the yields of the individual reactions, because you can always produce more of the parts. So initially the PCR, we used primers that we got off the iGEM website, the VR and VR2 primers. They didn't really work well, very poor yield, and we seemed to get quite a bit of non-specific products. So we went about this, we tried optimizing, but that didn't work out, so we made our own primers. Which worked very well. And the only thing is that because there's multiple different plasmids used by the parts, we actually had to have several sets of primers, which was a bit of a hindrance, but not so bad. So here we see different biobricks, different components on a gel. So the next step was to do digests. Obviously, to ligate the parts together, we need to digest the parts. And this also was troublesome. The digest would never really work very efficiently. And we suspected maybe it's because it's a PCR product. But as you can see in the diagram, our parts, which are flanked by the two sets of restriction sites, have more than 100 base pairs to the left and to the right of the restriction sites. So the restriction enzymes should have easy access to the parts, hypothetically. Because our whole idea is based around the use of GFP, we used a really simple assay, which was just use a UV light table used for looking at gels and GFP. And so we just grew some constitutive GFP expressing cells here, and it works great as far as we're concerned. And on to our results. So we constructed two different parts. One was an intermediate for our sender cells, where we put an RBS onto the aspartate producing enzyme, and the other was the entire construct for the receiver cells. And as you can see there, two. Oops. And here we have a picture from yesterday. of our first successful test of our construct, where basically we have our transformed cells growing on a regular agar dish, and we've added a little bit of AHL at a concentration of 10 millimolar in the middle, and it's diffused to actually quite a bit of the plate. We're surprised how quickly it diffused. And you can really readily see where the AHL has induced expression of GFP. So we're very happy that works. Now for next year, obviously we didn't get as much done as we wanted to, primarily because of difficulties with the actual construction. Now we want to increase the student pool. In the end, there was 4 of us working on this project, with 2 of us really in the lab. And so for next year, we really want to have a full-time commitment. During the summer, we had other things going on. This was sort of an aside. So we think that it's really necessary to have at least a few people who can be in the lab full-time. Also, an earlier start date would be great. We really only started in earnest in the lab at the beginning of August. And a broader student background, mostly it's bioinformatics, computer science. We could use some biologists, some biochemists, perhaps. Maybe. All right, and so there's actually a picture of our original team. Quite a few more people than you see here today. But we really want to thank some amazing people from the University of Calgary, Lisa Allen and Sonia Georgievic. They're the lab coordinators at the BHSC. Also, we want to thank Philippe Cecchini, Cynthia Collins, Dr. Mike Surratt. They're our advisors. And the Bachelor of Health Science program, which took us in and filled all our laboratory needs. Thank you. Yeah. Also, want to thank Christian Jacob, right there. Do we have questions? Yes, that's a very good point. We are concerned with the problem of mutations introduced, for example, by Taq polymerase. We actually used platinum Taq, which is a high-fidelity enzyme, to sort of mitigate that risk to avoid mutations. Yes?
So what gives rise in your simulations to the clustering in the 4 corners?
That is what gives rise to the clustering in the 4 corners we saw in the movies there. I think that is most likely an artifact of the computer simulation and the fact that it is a bounded world with 4 corners. So I doubt we would see anything like that on an actual petri plate.
It would probably be circular and
yeah. Any other questions? Would we use the same construction method again? I think we probably would. Now that we have the bugs worked out of the PCR method, it does seem to work fairly well. Is that right?
If there was a better method, we'd probably go for it. But the PCR, at least to generate the starting DNA, seems to be a very good way of doing it. Now, the main problem we really ran into in the end, and what limited our construction capacity, is the digests never really worked very well, and that really limits how well you can ligate parts together. But if there was a better, faster method, by all means, we'd use it.
Do you think that's a property of digestion, or do you think if the parts were better documented or better constructed,
better documentation of the parts would definitely be useful. We found that often the parts didn't really have all the information we would like. Probably sequencing would be also a good idea. We never got around to sequencing some of the parts. So there were some suspicions that maybe the restriction sites weren't exactly where we thought they were. Also, we had some issues with some of the parts where the sizes of the parts we actually had didn't match what was in the registry. So we of course know that due to our the primer sites, the parts are going to be about 200 base pairs longer than from the registry, but we were finding it was off by much more than that. Yes?
What's the time range of these experiments? Like, what's the time range from when you start the experiments and when they come into the
So your question has to do with how long it takes for chemotaxis and for them to sort of undergo this movement inwards and then back out in the simulations. Now I'm going to pass the mic over to Patrick because it's his realm. But in general, the one thing I can say from the lab is the induction of like AHL takes about an hour. After 30 minutes, you don't really see anything, but about an hour later, you see them completely on.
Yeah, for the induction of AHL, that's about all we can say. We never got to the point where we could really visualize the chemotaxis really clearly. So yeah, it would definitely be within 30 minutes or an hour of the entire experiment finishing. So we would have to measure whatever chemotaxis had occurred within that time.
In terms of the model?
In terms of the model.
Scales, maybe that's what they're asking.
Oh, what do you mean exactly? What do you mean exactly?
Well, I'm just saying in terms of the way you run the experiment, I was wondering how long it would take for that kind of thing to happen, because if you're monitoring it with the UV lamp, were you concerned that you're going to be mutating your cells as your experiment's going on, even if you're just giving a flash?
Right. The idea we had for that was to take our images at a time lapse. So we were going to set up a camera over top of the UV plate and take an image maybe every minute or every 2 minutes, every 30 seconds, something like that. That would allow us to keep the UV light off for most of the time. Yeah?
Do you have any idea like how many sender cells you need to produce enough AHL to produce that particular protein that where you can like fluoresce all the cells?
Do we have any idea of how many sender cells would be necessary? We don't have any hard data for sure. We didn't actually get to that point, but I imagine it wouldn't be a very great number. As we saw with our final plates, these cells are extremely sensitive to AHL. There is no gradient in their activation. It's very on or off, very binary. So I imagine not an enormous number, a fairly a thin lawn should be sufficient.
Yeah, the only one thing I want to add to that is that we were using 10 millimolar AHL, and we're adding minute amounts. Basically, we were dipping a pipette tip and then touching it to the plate, and we were actually surprised how far the AHL actually goes. Originally, our idea was sort of to once we make some sort of pattern with the light, that's sort of that's permeable, and so it'll diffuse even faster. So really what that says is maybe we don't really need high levels of expression in the sender cells.
How much time do you think it would take you to get this completely up and running and working?
If assembly, sorry, so how long would it take to fully implement the project as envisioned using the current techniques? Well, it really depends, I guess, on whether or not things work. If things worked, it's incredibly fast. You know, digest an hour, a ligation an hour, then 3 hours of PCR, and really a few weeks. But to really, you really have to repeat most of these things 3 times on average, probably the digest for sure. So I'd say 3 months. So for next summer.
The modeling system we used is, oh sorry, I should repeat. The question was, what modeling system did we use and what type of equations did you say?
Well, yeah, the efficient approach, I mean, it's partial differentials, more like simulation is not like how you just take some assumptions of the simulations.
So what kind of assumptions did we make for the simulation? What's that? Okay.
All right, I may answer this question. For our simulation, for our computer simulation, we take this swarm approach that I depicted in the first few slides, where we have tiny software agents that interact exactly as we would presume from our models. And in the software that you've seen, we actually have these tiny bits of software all interacting with each other, and they really take away the aspartate value from the different patches and stuff like that. So we don't want to have differential equations because we want to focus on the actual interactions. And as you have seen also, these different sliders that depicted our different parameters, how fast they eat away the aspartate or how fast they would move later on. They should be adjusted to whatever we saw in the theoretically in the experiments, and then we could also make some predictions about further possible setups. Does that answer your question? Okay, thanks.
Yeah. In the back there? Yeah.
Yes, you mentioned that you had to make different primers for each part.
So the question is, do we have to use different primers for each part? Not for each part, but some parts in some of the plasmids didn't have the primer sites. So we had two main primers that really worked for most of the parts. And actually we only needed different downstream primers. The upstream primer always worked. So we had, in the end, just 3 different primers that we used to generate all the parts. And they worked very reproducibly.
You mentioned sort of ideal team composition recruiting for next year.
Yes.
What ratio of biologists and computer scientists would you recommend for iGEM?
Okay, so the question was what ratio of biologists, computer scientists would we recommend for the team, and what would we do better for team composition? We realized pretty early in the year that our team was almost entirely composed of computer scientists, which is kind of a problem as far as doing biology wet lab work goes. So the ideal composition, I think, would be weighted more towards the biologists, I think, because the lab work is just so time intensive as a rule. Not even, no. There were 3 of us primarily who did work in the lab. Yeah, for simplicity was mainly the, the question was for the 2 signals, AHL and aspartate, would it be interesting to uncouple the 2 and treat them separately within the parts in the cell? And yeah, for the only reason that they were put on the same device to begin with was for simplicity's sake. But assuming we had a functional experiment, it would be interesting to switch out the ribosome binding site on one of them, for example, and change its expression and tune it that way. Or perhaps put them on completely different devices with different promoters that have different binding affinity. But we never did quite get to that stage. Did we submit a part in the end? I don't believe we have submitted anything yet, but there is one part we would like to.
Yeah, there's one
or
part
2.
we would like to submit for sure, and one intermediate part which we didn't really think would be worthwhile submitting, but we could submit those. Like we said, the actual working construct was made on Thursday at 1 in the morning. So. Sorry? Calgary, that's mountain. Okay. Yeah, well, thank you very much.